So at the end of the first section, we posed a number of different questions about antibiotics. The main one being, how is it that we sequence antibiotics? We're going to get to that, but let's take a detour to learn about another question, which is, how is it that bacteria make antibiotics? If we can learn about this, maybe we have some insights in terms of how to sequence them. So we described an antibiotic peptide as a mini protein. And the question then is, how is it that the cell produces proteins? So it starts at the level of DNA. This is one of the many reasons why DNA is so important. DNA is a double-stranded molecule, and it gets transcribed into RNA, which is a single-stranded molecule. So this is a complex molecular procedure that, for the most part, we're not going to get into. But in terms of strings, how we think about this is that RNA has the same bases or similar bases as DNA. And so the G for guanine, the A for adenine, and the C for cytosine are not going to change. However, the T, which represents thymine in DNA, gets represented instead by the base U, or uracil, in RNA. After this method, which is called transcription, DNA goes into RNA. RNA is then translated into peptides, or proteins. So we have 4 nucleotide bases: adenine, cytosine, guanine, and uracil. And I mentioned that there are 20 common amino acids. So here are the table of amino acids along with their 3-letter codes. And unlike the transcription of DNA into RNA, where we had 4 letters in each case, there's not a natural one-to-one correspondence here. So our question is, can we translate 2 nucleotides at a time? Could we take all tumors, AA, AC, AG, AU, and can we map those into proteins? The answer is going to be no, right? We don't have enough. There are going to be 16 of these tumor nucleotide pairs. So we're going to need at least 3 nucleotides at a time, and that's going to give us enough, okay? And so this motivates the definition of a codon, which is just a triplet of nucleotides. And the genetic code is the term that we use for the assignment of codons into single amino acids in order to make proteins. So here's an example of one codon that gets sent to one base. So tryptophan is always encoded by UGG. Usually what happens is that you see 6 different codons, or not necessarily 6 different, but multiple different codons. So here we have 6 different codons going into lysine, or L. You also have 3 special codons called stop codons. These serve as messages to halt translation. So they say, stop translation here, and don't continue translating. So these methods of transcription and translation, represented by beautiful molecular processes that we encourage you to learn more about, but that we're interested in on a computational level, form what are called the central dogma of molecular biology. So we start with DNA, it gets transcribed into RNA, that gets translated into protein. And this was a term, the central dogma of molecular biology. This term was coined in 1958 by Francis Crick of Watson and Crick fame. And I always thought dogma is kind of a strange word to use because dogma in English means a belief that cannot be questioned or overturned in any circumstance. And it's usually used in a religious context. And that's usually not a word that you see in science. And Crick admitted years later that he had made a mistake. He thought, he realized that he should have used a synonym that was more appropriate, like principle, but he used the wrong word, and now it's something that we're stuck with. And we should have a hint that because we've used this word, this strange word dogma, there may be some irony there, right? Maybe this dogma doesn't necessarily hold all the time. So assuming that it does hold, our goal is we know a lot about genomes, and if we have sequenced genomes, can we look for the mini protein of tyrosine B1 in the genome? So can we find a 30mer in this Bacillus brevis genome that transcribes into RNA and then translates into the peptide of length 10 that's represented by tyrosine B1? Unfortunately, there are going to be thousands of different 30mers that could do this. So I'll show 3 here, right? Notice that each of these 3 DNA strings gets translated into the same string of amino acids. And if we look at the 3 different DNA strings, we can notice that they're not very similar. So there are thousands of different ones, and they're not very similar, so we can't really draw conclusions by trying to reverse the process of translation. Another thing to notice is that translation is going to be able to start pretty much anywhere. So by anywhere, I mean it doesn't have to, if you look at this DNA string, this DNA string here gets transcribed into an RNA string. Similarly, its reverse complement gets transcribed into an RNA string running in the opposite direction. When you look at translation, though, translation can start at GUG, in which case the translated amino acid is valine. It could also start at U, though. So it could start and reading off UGA, in which case it's translated into a stop codon. And thirdly, it could be read as GAA, in which case it becomes translated into glutamine. So we wind up with 6 of these different reading frames, or ways of reading off the same strand of the same molecule of DNA, 3 from this strand running this direction, and 3 from the reverse complementary strand running in the opposite direction. We also have this additional fact that the tyrosine B1 peptide, like many peptides of its kind, is actually cyclic. So rather than just the one linear representation as a string of 10 amino acids that I've given you, we get 10 different representations depending on where we start on the string. So if we start at valine, we get one representation. If we start at lysine, we get a completely different one. So then we come back to our question and we say, how many 30mers in the genome of Bacillus brevis are actually going to encode one of these linear representations? So this is a question that we ask. And we could go off and program it in a computer and have the computer run our algorithm. And then our computer would say, well, actually, there aren't any 30mers in this genome that encode one of these linear representations. So that's weird. And our conclusion then is that somehow something weird is going on with this dogma, right? Somehow it's not getting translated from the DNA of Bacillus brevis. So to learn a little bit more about this process, I'll tell you that I mentioned that it's a beautiful molecular process, transcription and translation. Transcription is carried out by a molecule called RNA polymerase, and translation is carried out by an enzyme called a ribosome. An obvious idea then to say, is it the case that that an antibiotic peptide is maybe outside of this dogma, is to say, well, let's shut down the ribosome. If the ribosome is in charge of translation, let's shut it down and see if these mini proteins are still getting produced. So in 1963, that's what Edward Tatum does. He inhibits the ribosome in Bacillus brevis, and he hypothesized that all translation would stop. But instead, the production of some peptides, including the tyrosidines, like tyrosine B1, keeps going. All right, so this is a major sign that the dogma doesn't necessarily hold in all cases. 6 years later, Fritz Lippmann comes along and says, well, these tyrosidines are actually non-ribosomal peptides. So they're synthesized not by the ribosome, but by a completely different process that relies on a giant protein called NRP synthetase. Now, this giant protein is made up of a number of different modules. It's made up of 10 different modules, actually. And each module is responsible for adding a single amino acid to this small peptide of length 10. So it's a huge peptide made up of a number of different pieces, and each piece is responsible for just one amino acid. Once the amino acids are added, then the peptide is circularized. OK, so we've seen that the cyclic peptide tyrosine B1 is not produced based off of the genome. So we need to move beyond genome analysis to try and figure out what its sequence is. It's not going to be hidden in the genome. The tool that we're going to use is the workhorse tool of this approach, and it's called a mass spectrometer. Here's a photo of one that UCSD uses, and we're going to think of it as an expensive molecular scale. Now, this asks an important question, which is, how is it that we actually measure the mass of a molecule? So we're going to define a Dalton as the mass of a proton or a neutron, approximately. That's not an exact measurement, but it's approximately what a Dalton is. And the mass of a molecule, then, is going to be equal to the sum of the protons and neutrons and all of the atoms of the molecule. So let's look at an example. Let's compute the mass of glycine. So glycine is an amino acid, and it has chemical formula C2H3ON. All right, so it has 2 carbon atoms, and carbon has 12 protons and neutrons. So we do 12 times 2. Then we have 3 hydrogen atoms, each of mass 1. Then we have a single oxygen atom of mass 16 and a nitrogen atom of mass 14. So when we add these together, we get an approximate value of 57 Daltons. The actual mass is 57.02 Daltons, and that requires a more technical, precise definition of what a Dalton is. But we're going to use this rounded off approximation to an integer, and we're going to refer to this as the integer mass of glycine. So we can compute the integer mass of each amino acid that we have, and this gives us what we call the integer mass table. All right, so you'll notice we have the 20 amino acid masses beneath the one-letter abbreviation of each amino acid. So the question then is, well, what's the mass of something like tyrosine B1? We know the sequence of tyrosine B1, so we just need to go through and compute its mass as the sum of each individual amino acid. So V has mass 99, K has mass 128, L has mass 113, and so on. So we can go through, we simply add the constituent masses, and we get 1322. So that's the integer mass of this tyrosine B1 peptide. I'll note before we continue that 2 amino acid pairs, I and L, as well as K and Q, have the same mass. So when we weigh the molecules, we're not actually going to be able to distinguish between I and L or distinguish between K and Q. And so we're going to move essentially from an alphabet of 20 amino acids to an alphabet of just 18 integer masses. We'll work mainly with integers. Now in this talk, I'll show the actual amino acids in terms of their letters. But if you're working with them on a computer, you probably want to just assume I and L are the same and assume K and Q are the same. So let me show you then how this mass spectrometer works. So you're going to take multiple copies, a sample of this peptide, and you're going to plug them into the machine. And then the machine is going to chop them up into a lot of different pieces. It's going to blast them into fragments. And then essentially it's going to weigh these fragments. So this is an ideal scenario where it gave us the mass of every possible subpeptide or fragment of this NQEL peptide, circular peptide. And I'll highlight the masses because it doesn't tell us what the subpeptides are. It tells us what the masses are. Notice that here, 2 of the masses, 242, are equivalent. So we're going to call this the theoretical spectrum. The theoretical spectrum of a cyclic peptide is chop it up into all possible fragments, weigh those fragments, and then add 0, which is just an empty string, and 484, which is the mass of the entire peptide. So if you know the sequence of a peptide, computing its spectrum is just a trivial exercise. Chop it up into all pieces, use the integer mass table, and figure out what the mass of each fragment is. That's not bad. What about reversing it? What about going from the spectrum to the peptide? That's really the computational problem that we have. The mass spectrometer generates a spectrum, an experimental spectrum, and then we want to determine a peptide that came from it. That's going to be a harder problem. And we call this the cyclopeptide sequencing problem, so to reconstruct a cyclic peptide from its theoretical spectrum. So now we have a well-defined computational problem that's going to model what the mass spectrometer is telling us. And here we'll study a first idea of an algorithm for it. So first I'll tell you that the mass of the entire peptide is usually known. So we'll usually already know that tyrosine B1 has mass 1322. Our algorithm is going to then say, all right, let's generate every possible peptide of that mass. So in this case for tyrosine B1 it's 1322. Let's form the spectrum of every one of those peptides, and then let's look for matches with the given spectrum among those peptides. This is an example of a brute force algorithm. So in general, a brute force algorithm is going to try all possible solutions that satisfy a certain condition. So then our question is, to determine how long this is going to take, we ask how many peptides have a given integer mass. So in specific, how many peptides have integer mass equal to 1322? Here's a graph that shows the integer mass of a peptide versus the number of peptides that have that mass. And if we look at 1322, I've shown red lines here, we go up from 1322, and then we go over and we see it's almost at 10 to the 14th. So the answer is trillions of different peptides have this mass. And that's not going to be good. We don't want to have to consider trillions of different possibilities. So let's ask ourselves then, what is it that makes brute force so bad to begin with? Or at least this brute force algorithm. Some brute force algorithms may work well if they choose the correct condition. Here's a peptide, NQEL, right, that we're going to start working with, and another peptide, TMDH. So the total mass of NQEL is 484, and the total mass of TMDH is 484. But they're completely different, right? They don't have any of the same amino acids, and they're not going to have, so by extension, they're not going to have any of the same subpeptides. And so we need to find a way to exclude this completely. We don't want to ever have to encounter this peptide, all right? And if the way that we're going to get at that is to say, let's look at the spectra of these two peptides. So on the left here, I've shown the spectrum of TMDH, and on the right, I've shown the spectrum of NQEL. They completely disagree, right? So you may notice this. They agree at 0, and they agree at 484. And nowhere in between do they have the same two of the same masses. So we want to use this to our advantage. We want to use this to start designing an algorithm that's going to eliminate TMDH based off of its spectrum, which is not good. So our idea is, rather than try to form big peptides all at once, let's slowly build up our candidate solutions from smaller peptides. At each stage, we're going to test its spectrum and make sure it's on the right track. And if it's not on the right track, we're going to kick it out. So that's going to allow us to restrict the total number of peptides that are under consideration. OK, so now we have an idea for a different algorithm. And it's going to be based off of an approach called branch and bound. So let's learn a little bit about this. And I'm going to give a really hazy, vague overview of what a branch and bound algorithm's idea is. OK, so you're going to start with a simple candidate solution, and you're going to expand it or branch that out into different solutions. And then you might do this again. And then after you've branched out into a number of possible solutions or small seeds of larger possible solutions, then you're going to say, OK, can we exclude any of those? Can we eliminate any of these candidate solutions that we have? And that's called the bound step. All right, so from here to here, we've eliminated 5 solutions. And we're left with, these are not necessarily solutions, but they're at least candidates for solutions at the leaves of this tree. Then we say, OK, let's branch again. And then we'll need to bound. Based off of some condition, we'll exclude some of these edges when we move from branch to bound. And now we have maybe some candidate solutions, and we can go ahead and test these. So we've found a way of eliminating certain solutions before we construct them. So we want to apply this same idea to eliminating some candidate peptides before they ever get generated. All right, so let's apply it to cyclopeptide sequencing then. Here's a possible spectrum, all right? And our first question is, which amino acids have masses in the spectrum? So if we bring back this integer mass table, then we highlight P has mass 97, and it occurs in the spectrum. V has mass 99, it occurs in the spectrum. We also get T and C. So we're going to start with 4 1-mer peptides, all right? We're going to start with just these single amino acids. And what we're going to do is we're going to extend these 4 into all possible 2-mer peptides, so all possible amino acid strings of length 2. So we get PA, VA, TA, and CA. We get PC, VC, TC, and CC, and so on. All right? So we started with 4, and we're going to now multiply that by 18 because we're adding each possible amino acid onto each of these 4. And we're left with 72. So we want to know how we can trim this list. Right? So we've done our branch step, and now we need to do our bound step and bring it back. So to do this trimming, we'll notice that PV is what's called consistent with spectrum. Here's one possible tumor. And you'll notice that the mass of P is equal to 97. It's present. The mass of V, the other letter in PV, is present as 99. And the mass of this entire small linear peptide, PV, is 196, and it's also detected. So that's called consistent. When you chop up this candidate solution and weigh its fragments, that all those fragments are found in the spectrum. CD is one that we actually created in the expand step, but it's inconsistent with the spectrum. So although 103, the mass of C, is present, is detected, 115 and 218, the mass of D and the mass of the entire tumor peptide CD, they're not found. You can't find 115 or 218 in this spectrum. So we want to exclude CD. And after we do this step and we figure out what it is that we need to trim, we're left with 10 amino acid strings of length 2. From our 72, we've cut it down to 10. So let's do this exact same thing. Let's expand each of these in all 18 possible directions. We'll get now 180 different 3-mer peptides. And then let's check whether or not each one is consistent, and we'll trim it down. After we do this, we go from 180 to 15. And we say, OK, this is going well. We're preventing these peptides from growing crazily. And so we expand them, and then we'll trim. So we go from 15 and do 15 times 18, so we get 270. And then we find which of those 270 formers are consistent with the spectrum. And we're down to 10. We do it one more time, expand, and then trim. And we're left with 10 consistent 5-mers. All right? And you can verify that if each of these corresponds to the exact same cyclic peptide, and the spectrum of this cyclic peptide coincides exactly with our experimental spectrum. So now we have a method that's going to work, and it looks like it's going to be pretty efficient. I'll just summarize what it is. We find all the amino acids whose masses occur in the spectrum, and we add them to a list. Then we extend every peptide that's currently in the list in every possible direction. So we add each of 18 different amino acid masses onto the end of it. And then we have this trim step where we say, let's take which of these are inconsistent, and let's just get rid of them. Then we'll return any peptides whose theoretical spectra match the experimental spectrum that we're given. And we'll do steps 2 through 4 over and over again, this branching and bounding, and then testing until we run out of peptides to consider, until we have all inconsistent peptides. So we've seen it works really well on an example, but we ask ourselves whether or not this algorithm is efficient in practice. So the brute force algorithm that we saw before, it was exponential. And by exponential, I mean if you look at its input size, the number of steps that it has to take, or the number, in this case, the number of peptides that it has to consider, grows exponentially with respect to the input size. It may be true that the branch and bound algorithm that we've come up with is exponential on some data set. Like somewhere way out in the galaxy, you can form some data set that it takes a really, really long time to run, and it's prohibitive. But in practice, it's actually really fast. So it looks like we're done, and we can have a party and go home. That's the question, though. Can we really? And I'm going to have to pop that balloon and say no. OK, so at the end of this last section, I canceled our party and said that we have this algorithm that works really well in practice on theoretical spectra. But the issue is going to be that when we move to experimental spectra, we're going to have issues because these experimental spectra often produce errors. Here are the theoretical, and an experimental spectrum for the peptide NQEL. So the theoretical spectrum is on the top, and then we're giving you a hypothetical experimental spectrum for this same peptide. This experimental spectrum highlights a couple of different ways that the experimental spectrum can be flawed. So it can detect false masses. So these are masses that are present in the experimental spectrum, but that don't correspond to real masses in the theoretical spectrum. So examples are 99 and 299 here. We also have missing masses. So they're masses that are in the theoretical spectrum of the correct peptide, but that don't actually get produced in the experimental spectrum. So they're missing. This is a problem because our current implementation, in order for it to consider a peptide correct, the peptide's theoretical spectrum has to exactly match the experimental spectrum that we have, and that's clearly not the case here. So the question then is, what do we do? And what we're going to do is, instead of having to match the spectrum exactly, we're going to instead score a peptide based off of how similar it is to the experimental spectrum. So for example, here, right, the theoretical spectrum of NQEL is still pretty similar to the experimental spectrum that we have. Notice that it shares 11 masses. So we're going to say that the score then of NQEL with this experimental spectrum is equal to 11. So we need to use this scoring function to say certain peptides are better than others, and to use this to modify the branch and bound algorithm that we have in order to make it a little bit better. So the idea that I want to use here is similar to something called a cut in a golf tournament. Now, I'm a huge golf fan, so maybe I'm just partial to this idea, but I thought it fit it pretty well. So what the cut does is it reduces the field to only the players that are deemed to be in contention for the tournament. So if there are players that are not doing too well after a couple of rounds, then they say, okay, well, we're just going to let them go home, and we want to have the tournament only for the players who have a chance at actually winning the tournament. So here's a hypothetical leaderboard, and we may say in this hypothetical small tournament, we want to keep the top 3 players on the leaderboard. So keeping in mind that low golf scores are good, we put the cut line here. All right, but this would be unfair to this golfer, right? Because they're tied with the third place golfer. And so we need to say, well, we keep the top 3 players as well as ties, right? So we keep the top 3 players with ties. And so this is going to motivate the bound step that we do. So after this cut, we have 4 players remaining. So the new idea is we're going to call it leaderboard cyclopeptide sequencing. And so how it works is that we're going to start off with a 0 peptide. So this is an empty string, essentially. And then we're going to extend each peptide on the leaderboard in each of 18 possible different directions. So we add each of the 18 different amino acid masses to the peptides that we have currently in the leaderboard, which when we begin is empty, or it has just the 0 peptide. And then we're going to have a step, this cut step, which is the new bound step, and it's going to cut the low-scoring peptide. So it says, assign everything a score, right? See what the score of every peptide is, and then cut the low-scoring ones. Get them out of there so that we can prevent the number of solutions that we consider growing, and so that we can keep only the high-scoring peptides. So we're going to keep the top-end peptides with ties. Then the next step is going to say, okay, we'll update this leader peptide, right? We're going to keep a track of what the leader peptide should be, and if there is a higher scoring peptide that we can find on the leaderboard, so if something moves up and becomes a higher scoring peptide, and its mass is equal to that parent mass that we can detect. By the parent mass, I mean we said that we're always going to be able to know in advance what the correct mass of the entire peptide should be. So we always want to check if the candidate solution that we have has a mass that's equal to that total mass. And then we're going to eliminate any peptides whose mass is going to grow too big. So if their mass exceeds the parent mass, then we're not going to want to expand them anymore because their mass is too big and we want to get rid of them too. So once we do these steps, we're just going to iterate these steps over and over again. So we have a branch step, and then a bound step, and then we have an update step where we say, is there a leader? And can we get rid of anything whose mass has gotten too big? And so at the end, after we go through this procedure, we're going to branch and bound and branch and bound. It's actually going to guarantee to always have an empty leaderboard at the end. So that's at step 6, after we go through these steps 2 through 5, we're going to have an empty leaderboard. And then at that time, we return what is our leader? What's the best peptide that we've encountered so far? So we have this updated method, but I do want to give you the warning that it's what's called a heuristic. So because it's dealing with high-scoring peptides, there's a chance that there's an initially low-scoring peptide that winds up eventually being the highest-scoring peptide. So that eventually would be our leader, but because we cut it in an early stage, because we said it's not in the top however many N peptides, then we throw it out. And so there is a possibility that we throw out the correct peptide, or what will eventually become the correct peptide, at the expense of having a quick method. And so for this reason, it's a heuristic. Let's test it, though, because remember, we're considering how well these algorithms work in practice. So here's a hypothetical spectrum. It has 10% false and missing masses. So I'll highlight where the false and missing masses are. So here are the false masses, and here are the missing masses. Now, we're not going to know which of these are false and missing to begin with. In particular, we're not going to have the missing masses present. That's the whole point. They're not present in the experimental spectrum. So we'll have this picture, but we won't know which of the masses should be green. And then so we say, let's run our algorithm. Let's run our leaderboard algorithm on this hypothetical data set and see what it turns out. And so it returns the correct tyrosine B1 peptide, which is where this came from. So we're happy. So then we say, well, maybe 10% false and missing masses, maybe that was a little bit optimistic. So let's introduce a slightly noisier tyrosine B1 spectrum that has 25% false and missing masses. And again, I'll show you, even though we won't know this, I'll show you where the false and missing masses are so you can believe me. And we say we won't know which masses are missing. And now when we run that same algorithm on this noisier spectrum, we're going to see that the peptide that it produces is actually not tyrosine B1. It's close, right? But instead of the correct amino acid here, it's been replaced with an AD. So this is bad. In this case, we wouldn't be able to reconstruct the correct peptide. So in the last section, we saw that our new algorithm, based off of the leaderboard and cut method, worked well on a spectrum that was close to the correct theoretical spectrum. But once we had 25% false and missing masses, it didn't reconstruct the correct tyrosine B1 peptide. I want to add some more bad news to that in this section, which is that these NRPs, these non-ribosomal peptides, including tyrosine B1, they in practice will contain more non-standard amino acids that are outside of the standard genetic code. And this is because they're outside of the central dogma. So for example, here is a relative of tyrosine B1 called tyrosine B. And it's very similar to tyrosine B1, except you'll notice that in this position it has an amino acid called ornithine. And this is a non-standard one. And so to deal with these non-standard amino acids, because there are so many of them, what bioinformaticians are assuming sometimes, and what we'll assume here, is that any integer between 57 and 200 can act as the mass of an amino acid. Right? So we've expanded now our integer mass alphabet from 18 masses to 144 amino acids by making this assumption. So then our question is, okay, we expand our alphabet, and now we have to deal with a larger class of peptides. Let's run our leaderboard cyclopeptide sequencing algorithm on this expanded alphabet to this spectrum 10 with 10% false and missing masses. Now remember, before, when we assumed the standard alphabet, it reconstructed the tyrosine B1 correctly. But here, when we expand the alphabet out to 144 amino acids, the algorithm now identifies the highest scoring peptide as this peptide, and it incorporates 98 and 65 into the peptide. So it doesn't reconstruct the peptide correctly. And notice that it's brought in two non-standard amino acids. The key point here is that although tyrosine B1 doesn't actually contain any of these non-standard amino acids, we won't know that when we're doing sequencing experiments. So we need to take them into account. And in this case, the algorithm would tell us that it has non-standard amino acids in it, and that's not good, because it doesn't. So we've now seen that the algorithm that we have, this branch and bound algorithm, is not going to perform well on noisy data sets. And as soon as we go from 18 amino acid masses to 144, we're going to start to really struggle with using this algorithm. We're not going to give up on it. We just need to kind of figure out if we can find more information that's in that spectrum. Is there something that the spectrum is telling us that we just haven't been listening to? So our goal here is to say we've got to get the number of amino acid masses that we consider down. We've got to reduce these. So we've got to go from 144. Wouldn't it be nice to knock that down to 50 or 20 or even 10 in a great scenario? So let's recall this spectrum that we had, this hypothetical experimental spectrum. And let's just see what we notice about it. This is a great problem-solving strategy where if you're stuck on a solution, one of the most common things to do is consider a small example and look and see what that small example is telling you and see if that maybe generalizes. So here's our small example. It's just going to be for a peptide of length 4. And we're going to say, OK, well, we can't detect the mass of E. It's not in this experimental spectrum. So we're not going to identify that E is one of our letters, one of our amino acids, that way. But hold on. We do detect 257, which is the mass of QE, and we do detect 128, which is the mass of Q. All right? And if you subtract these two masses, you should get the mass of E. Right? E is what's left over with QE and Q. So when you subtract them, you get 129. So that's good. Can we find it anywhere else? The answer is yes. Okay? ELN is another subpeptide of this cyclic peptide NQEL, as is LN. And we notice that the masses of these two are in the spectrum as well. We get 356 minus 227, and that gives us 129. We even get one more. The mass of the entire peptide is 484. And the mass of LNQ, the subpeptide that doesn't include E, is 355. And when we subtract those, we get 129. So 129 is still there, right? It may not be in the spectrum, but if we start subtracting masses that are in the spectrum, then we're going to get 3 occurrences of 129. This is going to motivate our definition of what's called a spectral convolution, so the convolution of a spectrum. And it's the positive difference between every pair of masses in the spectrum. So here is the same spectrum that we had. We represent the masses along here and along here. And we go through and we subtract every pair. So we have 227 here is the mass of Ln. We don't know it's the mass of Ln, but we have 227. And here we have the mass detected of 370. And when we subtract them, we get 143. And we can do this for every pair, and we form this table. Now, I've highlighted certain masses, and I'll tell you why I've done that. The reason is we want to ask what are the most frequent elements between 57 and 200? Because remember, with E, we picked it up in a number of different places. We picked it up 3 different times. So we want to ask ourselves, what are the most frequent elements between 57 and 200? And there's a good chance that's going to give us what the amino acids should be. The 5 most frequent elements that I highlighted are 99, 113, 114, 128, and 129. And when we convert those from the integer mass table, we see that that's V, L, N, Q, and E, right? They correspond to those letters. So if we take the 5 most frequent elements, 4 of them are going to give us the amino acids of NQEL. So this is really promising on this small example. So now we have the outline of an algorithm called convolution cyclopeptide sequencing. And the first thing that it's going to do is to say, OK, given our experimental spectrum, let's form the spectral convolution of it. Then we're going to look at that spectral convolution and say, what are the most frequent elements in it? And so we may choose this parameter, capital M, taking the M most frequent elements between 57 and 200. So M may be 50 or 20 in practice. And then we're going to run our leaderboard algorithm using only those top M masses that we found between 57 and 200 in the spectral convolution. And we're going to form our peptides only on those integer masses. The question then is, does this algorithm really work? That's the question that we constantly ask, right? And so let's consider spectrum 10. Remember, when we expanded our alphabet, the leaderboard method didn't work for spectrum 10. So let's see if it works for spectrum 10 now that we're dealing with the convolution. So the first step is take the convolution. Then we pick the 10 most frequent elements in that convolution, which I'm showing here on the right. Most of these are actually standard masses, or correspond to standard amino acids. So 145 was non-standard, but the other ones were identified as standard. Then we run the algorithm on only these amino acids. So these are the only amino acids that will be allowed in our peptide. And the winning peptide is tyrosine B1, and we should be happy. So it reconstructed the correct peptide just from taking the 10 most frequent elements in the spectral convolution. So it worked really, really well. Well, then we ask ourselves, OK, what about spectrum 25? So what about this noisier spectrum that none of our algorithms has been able to correctly reconstruct tyrosine B1 from? What happens if we apply convolution cyclopeptide sequencing to that really noisy spectrum? And when we do it, it's good news. We can reconstruct tyrosine B1 again, and so now we can start the party. But I do want to mention one more thing about practical considerations, and I'll do that in the next section. OK, so we have this convolution cyclopeptide sequencing algorithm, and it works really well on the hypothetical spectra that we've considered. But these spectra aren't necessarily representative of spectra that we might encounter in practice. So in particular, we had 25% false and missing masses. And that's much less noisy than what we will be able to obtain from practical spectra. Also, I've said that the mass spectrometer is we can think of it as a scale, but it doesn't really just weigh the peptide fragments. It's a lot more complicated than that. What it does is, first it ionizes the peptide fragments, so it converts them into ions with a positive or negative charge, and then it sorts these fragments using an electromagnetic field. Once it does that, it's then able to not measure the mass, but the mass-charge ratio of each fragment. Then it determines the intensity, or approximately, we'll think about it as the number of ions at each mass charge ratio value. And so after it determines this intensity at each mass charge ratio value, we're able to create a graph from this. And so this is really what the spectrum is. The spectrum is a graph of the intensity on the y-axis and the mass charge ratio on the x-axis. So it's detecting the number, give or take, the number of ions at each mass charge ratio value. And from this, we somehow need to convert it into mass, right? All we have is mass charge ratio. So these, we want to consider the most popular elements in this, or the most intense peaks is what is the term that we'll use. And the challenge is now to reconstruct, in this case, to first convert to mass and then use those masses to reconstruct the peptide. In a case where we may not have 25% false and missing masses, we may have like 2/3 of the masses that are detected in our spectrum may be false, in which case we've got to really switch up what we do, and we may need to adapt the algorithms that we have. And so on STEPIC, this is where we give you the example to take a real spectrum and try to process it and adapt the algorithms that you have, maybe come up with new ideas, and try to sequence a real peptide from a real experimental spectrum.